Okay, hello everyone. Welcome back after the summer here back at the Broad or online. So welcome to this joint Broad MIT EECS Colloquium that is hosted by the Eric and Wendy Schmidt Center and AI+D within MIT. And so in this Colloquium series, I know I see many repeat visitors, so you already know. So we're hosting speakers who not only bring machine learning into the biomedical sciences to answer important questions there, but also go the other way around of taking biological or biomedical questions and then having them motivate new foundations in machine learning. And so we'll have like a really great talk today here exactly in that vein by Marinka Zitnik. She's a faculty at Harvard in the Department of Biomedical Informatics and also at the Kavli Institute. Not Kavli. Kavli is the award that you, Kempner, sorry. Kempner Institute at Harvard. I will talk about the Kavli Award because you were just awarded that. I was very excited to see it. So she was just named Kavli Fellow by the US National Academy of Sciences, which is really exciting. Congratulations. So she did her postdoc at Stanford and then got a bachelor's and PhD in computer science from the University of Ljubljana and then joined Harvard University in 2019. So her research is very much in line of this series, where she investigates the foundations of AI and machine learning to really enhance biological discovery, and then also go all the way to medical applications, to personalized medical applications and for diagnostics and treatment. And so I think we'll see a lot of that today in your talk. So Marinka, the floor is all yours. So thank you very much for being here.
Hi, everyone. Very excited to be here with you today. And thank you so much, Caroline, for inviting me to speak here, as well as for this kind introduction. So today I will talk about novel AI models, and particularly models that can be trained across a broad range of tasks to have the capabilities that can help us advance therapeutic science, particularly design of novel therapeutics and modeling of treatments in human populations. And throughout the talk, I will discuss and highlight various projects that all are associated with code bases and data sets that we are publicly releasing for the community, together with tutorials and documentations that are all linked from our lab website. So you are encouraged and welcome to check those resources later if you're interested in models and topics that I'll be describing. So in the recent decade, we are seeing increasing adoption of AI and advanced computational models across different disciplines in the scientific enterprise. And increasingly, AI is being closely integrated into scientific and research workflows across different stages of nearly every research project. And that is exciting and creates a lot of new opportunities, including challenges that we need to think about, where we are using AI to collect and process very large data sets, where we are using these models to generate novel scientific or biomedical hypotheses, and then where those hypotheses that are generated by computational models are then coupled with either simulations or experiments and validated in order to produce new scientific insights. And in particular, we're seeing this entire process and some of the fundamental questions that relate to the way the science is being done and the way the scientific method is applied across different scientific disciplines is being fundamentally affected by this new class of models and new class of algorithms that can help enhance and augment the work of scientists in order to produce new insights. And so here I'm just highlighting a couple of big advances or very interesting models that were developed over the last few years across different areas of biology and medicine that allow us now to generate near atom level accuracy structures of proteins and predict those structures from primary amino acid sequences, or screen really large chemical libraries with billions of compounds in order to find what are those compounds that might likely have certain desirable properties and potentially be a promising therapeutic candidate. To be modeling from the structural perspective of how do candidate therapies or such as small molecules bind actually and affect their therapeutic targets such as proteins and then exert therapeutic effect to treat the disease. And various other questions that go across biological discovery as well as medical applications that have an impact on real life in terms of patients in terms of providing early and accurate diagnosis for diseases that are otherwise very hard to recognize by frontline clinicians, or even then matching patients that once they're diagnosed with specific therapies. And so all this work over the last decade or so has been driven and in many ways enabled by some of the algorithmic breakthroughs over the last decade. And so what I'm showing here on this slide are three big areas of algorithmic innovation that have been productively used across the biomedical discovery. So the first, and I'm very excited about because it's closely related to my personal research interest and that of my group, relate to the idea of geometric learning, which is the paradigm concerned with developing models that can, by design, be trained on data sets with underlying geometric or relational structures. And so those kind of models have been in the recent few years productively used for problems in genomic medicine, drug discovery and design, as well as some structured EHR and healthcare-related data sets. The second key paradigm that is very important, especially for problems at the frontier of discovery, is that of self-supervised learning, which fundamentally allows us to be able to train larger models on data sets that do not come with explicit labels. And so that is important in settings where human annotation and label assignment is resource-intensive, expensive, or in some cases not feasible. And this paradigm of self-supervised learning has been productively used and been successful in various studies related to biomedical imaging and processing of natural language text, as well as biological sequences. And finally, and most recently, we're seeing increasing interest in models that can not only make predictions, but can also produce new designs in the form of structures or other elements that can then be, once generated by AI models, then synthesized in the lab and further refined. And this process of generative models has been successfully or is investigated and examined across a broad spectrum of applications, ranging from design of and optimization and refinement of molecular structures, all the way to designing chatbots or co-pilots that can help scientists with programming, ideation of novel ideas, and kind of generating interesting hypotheses. For the purpose of this talk, I will focus mainly on one important capability or increasing interest in this area, which is that of how to make models broadly generalizable, how to move away from designing a single model for a single data set and optimize it for a single task, and kind of move towards models that can be trained on a vast data at scale and then easily adapted to a broad set of downstream tasks. And in the literature, those models are now typically referred as foundation models, sometimes also general purpose models or generalist models. And their motivation for the problems that I will be describing in this talk is the following. For many problems in the area of therapeutic science, drug discovery, and development that we're interested in, there typically is not necessarily just a single data set or a single biological scale that captures the full complexity of the underlying phenomenon that we're studying. So biomedical data then really need to be integrated across multiple scales and often multiple data modalities. So the foundation models broadly then allow us to design models in such a way that we can bring together this data across different modalities and biological scales and train a single model with a unified objective that is based on self-supervised learning without requiring labels for any of those data types, and then have a single model that is trained in a consistent manner. And the reason why we would want to do that is because once trained, those models can be adapted to a vast array of downstream prediction tasks. And the really hope and the goal of our overarching goal of designing those models is that we want to design them in such a way that when there are introduced a new data set or a new task, then the model would be able to produce high quality results with no or minimal training. And so there is lots of effort put into training these models such that once trained, it can be easily adapted to the broad range of tasks. And for the purpose of this talk today, I will focus on two aspects of foundation models that we are working on in my group. In the first part, I will talk about knowledge graph models. And knowledge graph models are models that allow us to train models on unified knowledge graph or heterogeneous graph representations. And I will describe a model that we developed that can process a number of therapeutic tasks in a unified formulation. In the second part of the task, this talk today, I will talk about contextualized models. And contextualization is a problem that I believe is one of the key problems that we need to think about and address if we want to make sure that predictions produced by our models are more actionable in the sense that they can more easily translate to downstream experimental investigation by making sure that the predictions made by models can be adapted or adjusted to biological context in which the model operates. For example, that context can be given by single cell types, it can be given by tissue context or disease states. So let's start with the first part, that of knowledge graph models. And so what we will be interested in here, and I will describe, is a model that can implement this idea of foundation models for the problem of therapeutic use prediction. And specifically, here we have built a large knowledge graph that summarizes a molecular understanding of 17,000 clinically recognized diseases and the entire spectrum of FDA-approved therapeutics nowadays, and represents all that data into a single data structure that is referred to as a knowledge graph, which you can think of it as a graph or network where we have nodes that represents different biological entities or biomedical concepts, and edges or relations between those nodes in the graph carry different semantic meaning. And once we have that unified data representation, a model called TXGNN, which stands for Therapeutics Graph Neural Network, is trained on that network and then can process a number of therapeutic tasks related to predicting candidate indications, which are potential positive treatment effects of drugs, as well as contraindications referring to the situation where a certain drug should not be prescribed and given to a patient because it can make their health worse. And it can do so in a unified formulation. So why do we need these kind of models, and why do we want this? We can benefit from models that are trained across such a large number of diseases. And so we were particularly motivated by the question of being able to develop models that can help recommend therapeutic opportunities for those diseases that have very few treatments. And the way this is technically achieved nowadays is through the capability known as zero-shot learning, which allows these models to make predictions, in particularly to identify and recommend drugs for diseases that currently have no treatments at all. Why would we even want to do that? There are various reasons, and I will mention two big motivating factors that were guiding principles for us. First is that there really are no effective or even FDA-approved therapies for many complex diseases and for most rare diseases. In particularly, there are over 7,000 recognized rare diseases. Rare disease in the US being defined as disease that affects less than 200,000 individuals per year. However, collectively, there are so many of those diseases that the number of individuals affected by them is considerable. However, only 5% of rare diseases have any FDA-approved drugs. And even for diseases that have approved therapies, one would be interested often in finding new drugs as alternative options that can be used for patients for which existing treatments do not work or lead to severe side effects. And second is that the second big factor is if one can effectively find a way to take an existing drug or a drug that is already on late stage of drug development and expand its use so to diseases that it was not initially designed for in this drug repurposing fashion, then that can lead to much faster translation of that in predicted indication to the clinic, which is associated with lower development cost. The problem, however, is that most of these drug repurposing opportunities were really accidental findings over the last several decades. So the question becomes, can we develop models that can identify these drug repurposing opportunities at scale? And with a particular focus that we want these models to provide useful hypotheses for diseases with few or no existing therapies. And that's what TXGNN was specifically designed for. It was driven by this problem that we want to have the model and develop a model that has zero-shot drug repurposing capability, which is in sharp contrast to what standard supervised models can do. So the first part to do that is to kind of build a data set and think about the representation that allows to bring very different types of data together in a single data set that can be then used as input to train a self-supervised model. And to that, we built a knowledge graph that we refer to as PRIME-KG. And PRIME-KG is a precision medicine-oriented knowledge graph that can be thought of as heterogeneous graphs that I previously described that contains information on diseases, phenotypes, genes, proteins, and their known information about the involvement in the mechanism of action of drugs, as well as information about biological and molecular pathways. It was built with a specific goal in mind that we want to have a broad representation of diseases. And so that's one of the big focus of PRIME-KG. PRIME-KG is publicly available, and it's also can be easily automatically updated when new data become available. And so that allows us then to continually update both the data and the underlying model as new data become available. Once we have that PRIME-KG knowledge graph, then TXGNN model is trained in a self-supervised manner, and I will explain what that means for us in the next slide. But at the very high level, you can think of this model in the following way. This model, once trained, will take as input a particular query drug and its available information about the candidate mechanism of action of that drug and the description of the disease. And given these two elements and their associated data as input, the model will be predicting the likelihood of that drug being indicated or that drug being contraindicated for the phenotype and the disease that we're interested in modeling. And finally, a second component of the model will be extracting specific predictive paths from the knowledge graph that can serve and provide evidence for why the model has made a specific prediction. TXGNN is a graph neural network, so I want to just give a single kind of slight background on what graph neural networks are about. And so, as the name implies, graph neural networks are neural networks that are trained on graph structured data, such as this PRIME-KG knowledge graph. And fundamentally, you can think of those neural networks as multi-layer nonlinear transformation functions that need to be specified in such a way that for every element of the graph, which can be a node or a subgraph or an edge or even entire graph, that element of the graph on the left, shown as input, is mapped by a graph neural network, here denoted F, to some latent representation or embedding in the latent space. And the way graph neural network generates those latent representations is such that for two nodes in the input graph, if they have similar local neighborhoods, then we want that those two nodes have similar or embeddings in the latent space. And now there is, well, okay, great, the slides are back. And now the main question that you might be thinking about now, beyond what just happened, is that of, well, how do we know when two nodes have similar local neighborhoods? And now this is really big, important question, and that led to the entire field of research concerned with quantifying in various different ways and deciding where two nodes in the graph on the left have similar local neighborhoods. One of the simplest answers would be, well, if they are directly connected and they are proximal in the graph, they would have similar local neighborhoods. Or if perhaps their one-hop or two-hop neighborhoods overlap and in a major way, then they would be declared similar in the graph, and we would want to optimize the model such that predicting that embeddings generated for those two nodes would be highly similar, such as having high similarity between the vectors. And so the entire field of graph neural networks is concerned with specifying these functions that are then optimized through a series of neural message passing strategies that effectively learn what is a way to propagate neural messages, which are these hidden layer embeddings along edges of the graph, and aggregate them at every node, such that the final embeddings produced by a GNN model would reflect the topology of the input graph. And so TXGNN is such a graph neural network, but it's trained on a prime KG knowledge graph. The intuition why we would want to train a graph neural network on a knowledge graph in order to extract and derive drug repurposing hypothesis is that of prior knowledge in biology that kind of shows that drugs that present effective treatments for the diseases tend to have therapeutic targets that are in biological network close to genes and proteins that are associated with diseases. And so the way we think of this problem is in the following manner. So assume that we start and we have a specific therapeutic candidate for which we know what are the targets of that drug. It might be a small molecule drug that binds to a certain number of protein targets. Well, those protein targets do not act independently of each other in human cell. In fact, they interact with other proteins within the underlying protein and interaction networks. So we can expect that those therapeutic effects will actually propagate throughout the underlying biological networks and eventually reach other nodes in that biological network that are associated with the disease. And so graph neural network is effectively matching these specific patterns that summarize mechanism of action of a drug with known information about variant genes and proteins that are associated with the disease, and learns the relationships in such a way that it can then be productively applied to novel drugs and diseases in prioritized candidate therapies. So ultimately, the task that TXGNN is being optimized for is that of taking a subset of a biological network that consists of molecules and interactions that are dysregulated in a disease and finding, well, what is the therapeutic candidate that would reverse the effect of that dysregulation and kind of reverse the effect of the disease. And so that's what TXGNN can do. The specific novel component of the model is that of disease pooling. And disease pooling is a component that allows us to transfer the principle that I described from one disease to the other and effectively score what is the similarity between different diseases, so that for a disease for which the understanding of what molecules are being dysregulated and in what way in that specific disease, even if we don't have that information, we can, based on phenotypical description of that disease, relate the disease to other diseases and pull information from those other diseases to this poorly described disease in the knowledge graph. So what the disease model pooling can do is find for a specific query disease, identify in which other diseases in the knowledge graph are similar to the query disease, and then transfer information from those other diseases to this disease in question. And this is important for the model to have the ability to be able to provide accurate predictions for those diseases for which data are incomplete and highly sparse. So the specific module shown here on the slide, and specifically the right part of the slide, let me see if this works, is you can think of it in the following manner. So I'll just describe. On the bottom left part is a snapshot of prime KG knowledge graph that shows two local neighborhoods that are centered around one specific drug and disease node, shown in yellow in this radial patch and then in blue. And these local neighborhoods of drugs and diseases are being passed through a graph neural network that then generates for a query drug and disease their associated embeddings. And then the disease pooling module calculates a similarity between the query disease and the embeddings of other diseases in the knowledge graph. And based on those signatures for other diseases, it can easily and quickly retrieve what are other diseases that have most similar signatures. And those signatures from other diseases are then aggregated with the signature and embedding of the query disease through a gating mechanism in order to finally produce a high-quality, rich description of a disease against which drug embedding can be compared and final prediction can be produced. So this disease pooling module is really what drives the model to make to be able to handle then diseases for which data are really poor. Well, if we want to convince ourselves that this is indeed the case, here are some of analysis and experiments that we have done to check whether TXGNN can perform well in those and predict well therapeutic opportunities for those diseases with limited information available. Existing work and research today in this field is mainly concerned with identifying drug repurposing opportunities for diseases that already have some approved treatment or already have well-understood mechanism. And for those kind of tasks, it's actually quite easy for existing models to perform well because they often rely on the underlying notions of scoring similarity between known and approved drugs for a disease, and then finding what other drugs based on structural or mode of action type of information are similar to known and approved drugs in order to make prediction. This is not possible in the zero-shot prediction setting where for a specific disease there are no existing drugs and there is very limited mechanism and understanding of the mechanism, meaning that its local neighborhood around the disease is very sparse and they're incomplete. And so we wanted to design a series of challenges or data set splits that allow us then to evaluate in a systematic manner how well TXGNN can perform for those diseases. And so I'm showing the results where we split the 17,000 diseases into two broad sets. And so one broad set was that of diseases that have many or some known treatments and some molecular understanding, and those are the easy to predict diseases. And then diseases with poor molecular understanding or known FDA-approved therapies that are hard to predict. And on the right part of the slide, we see performance of TXGNN model relative to state-of-the-art graph neural network under these different scenarios. So what's interesting here to see that when we look at TXGNN model versus state-of-the-art graph neural network on the standard random data set split, performance improvement is relatively modest. But we see considerable improvement in performance where the diseases that are in the test set are those diseases that the model has never encountered in the training set because for them there were no available information given. And we can perform this analysis in numerous different ways. So one of the experiments that we have, empirical experiments that we have performed has constructed these data set splits based on disease areas, where all diseases within a specific therapeutic area were in the training set of the model, and the model did not encounter any of them in the test set and vice versa. And so here, for example, where the disease, one of the disease splits was concerned on adrenal gland diseases. And for that disease split, we see the considerable improvement of TXGNN across various other baselines that are based on recently developed heterogeneous graph transformers, attention-based graph models, as well as some of the language models. In this specific case, the diseases in the adrenal gland group or adrenal gland diseases were all in the test set and none of them were in the training set. And the model was challenged to generalize from other disease areas to adrenal gland diseases. And when we consider different disease areas, for example, here I'm showing 4 of those splits. We have many more in the paper that are concerned also with anemia or mental health diseases or cancer diseases. In all of those cases, the model showed considerably better generalizability when it was asked to solve and make predictions for diseases that were not encountered before during training of the model. Beyond benchmarking, we were interested in knowing whether the model can make novel predictions and whether those novel predictions can likely represent promising therapeutic candidates. So we have done numerous analyses of that kind, and I'm showing here 3 brief highlights. So one of the analyses that we perform is that of checking whether novel predictions made by the model are consistent or enriched in off-label prescription decisions that are made by clinicians in a large healthcare system. So here we work with the data on 1.2 million patients from Mount Sinai Hospital system in New York that was processed and split across 480 diseases and 1,200 drugs. And what we were interested in knowing is whether those predictions of novel indications that the model made that are not yet FDA approved would be enriched and found more frequently than expected by random chance in EHR records of patients where those drugs were prescribed in an off-label manner. And so what the summary result shows on the right part of the slide, it shows that the enrichment of novel predictions that were top-ranked predictions by the model. So if we look at top-ranked, top 1 prediction made for every disease, or top 5, or top 5% predicted drugs for a disease, we see an enrichment that is increasingly close to the enrichment of real FDA-approved indications, whereas if you look at the bottom of the ranked list that contained predicted drugs that the model was most confident that they are not promising, those drugs at the bottom of the list were actually had an enrichment score that is comparable to the enrichment score of existing contraindications, so settings where drugs should not be prescribed and given to a patient with a specific disease. We also can perform various temporal splits where we log the model to a specific point in time and then wait for a while and then test the model on predictions or discoveries that were made since the time the model was logged. So in this specific case, we retrained the model from scratch on all data generated up to June of 21, and then test the model against recent FDA approvals since then. And that is just one way of doing the validation. There are numerous other experiments that one can perform also to consider kind of splits at the different points in time to make sure that there is limited or no data leakage in terms of prior research on a specific drug candidate being included indirectly in the knowledge graph. And finally, for some of most promising predictions involving emerging pathogens, we have had a chance to share those predictions with our collaborators at Harvard and Boston University, who have looked at top-ranked predictions made by the model for an emerging pathogen and compared and used those top-ranked predictions as a guiding principle for screening in the wet lab setting. And what we have seen in doing so is that the yield of drugs that have inhibited the growth of the pathogen in the lab was an order of magnitude better than what would be possible to achieve if that screening would not be guided by AI model at all. And so all these various benchmarking and evaluation and analysis that is being done brings us to an obvious next question, which is how can we think about transitioning AI models to then real-world implementation and real-world deployment? And so here, let me share with you one specific story and experience that I have had in doing so with TXGNN model. So far, what we looked at is mainly benchmarking and evaluation of predictions that the model made in the form of predicted ranked drug list for a specific description of a disease. But what is important to understand is to what extent the model is also able to identify what are sets of biological interactions and the molecules involved on that path that contribute to the model making a specific prediction. So my lab has done quite a bit of research in this question of explainability and interpretability using counterfactual inference to develop models such as GNN explainer and more recently other explainability techniques that can, once the model is trained, extract parts of the input graph the models are trained on that affect prediction the most. And so we have implemented this functionality also for TXGNN model, and then we're interested in checking whether those explanations matched with predictions together provide value for our end users. So we performed a user-centric study where we have recruited a number of clinicians and clinical researchers, and we're wondering in knowing whether they can benefit from predictions and explanations that TXGNN provide, and whether they are able to more accurately and quickly decide and understand how the model works and whether predictions made by the model are to be trusted. So one of the important elements of this study was to ask a question around how do we actually present those AI outputs to end users? And this question touches upon HCI and data visualization and is often ignored by AI and machine learning community. And specifically here, what we were interested in knowing is what is an effective way to visualize and present those explanations. And we considered 3 different ways to visualize explanations. One explanation is that, the first one is that of neighboring nodes, where for a drug disease prediction that was made by the model, we really present and visualize to the end user their specific directly interacting neighbors in the prime KG knowledge graph. The second is that of subgraph-based visualization, where we are not only looking at the direct neighbors and their local proximity of drugs and diseases, but also at the entire subgraph that connects the drugs and diseases in the knowledge graph. And the third visualization is that of meta-path-based visualization, where we show these linear paths that start in a specific drug node in the knowledge graph and end in a disease node in the knowledge graph and go through a series of other biological entities and their interactions that then the end user can go through. And the first interesting result that we found was that path-based explanations have been considerably preferred by end users over node-based or these neighbor-based explanations and subgraph-based explanations. And that was across multiple usability metrics, including that of end user accuracy, which was a kind of test where we matched the domain expert in a specific disease area and actually query them whether they can guess whether a specific indication is actually a real indication based on predictions alone, as well as measuring the confidence of experts in the reliability of prediction the model made and the time it took them to navigate the predictions. Furthermore, when we evaluate and compare TXGNN to other baselines that do not consider and provide explanations, we see significant improvement in both accuracy and confidence of domain experts when we are designing the visual interface through which end users interact with AI model in a way that takes into consideration their preferences and expertise. So this model is currently online, and a version of it is available for everyone to use here as well at tixgnn.org. And it allows you to go to that interface and then start exploring some of the predictions and the systematic level predictions, particularly that do not expose the patient level data in a way that does not require any a priori registration and access. So beyond questions of designing visual interfaces and other kinds of ways that allow the end users to interact with AI models, I think that recent advances in AI create opportunities to think about interfaces that allow experts to query AI models in a more open-ended manner. And so in order for us to be able to do that, and specifically working with graph and graph-based modalities, it is often a data modality that is challenging for human users to directly navigate and interact because graphs are so massive and impossible to visualize in total. So the enticing alternative or exciting opportunity then becomes that of bringing and integrating text information into those models. And so we've been recently embarked on this question, specifically in the context of large language models that were primarily designed for in natural language sequences. And we're started to ask questions, what are the limitations or assumptions that were encoded into these models designed initially for natural language, and such as English text or text in other natural languages, and whether those assumptions still apply where we are transferring or using those models in the context of more general sequences or biological sequences, such as amino acid sequences of proteins. And so that has led us to the question of whether language models that are derived mainly from natural language processing field and are based on the language modeling objective of hiding a specific token and then asking the model to guess what is the token that was just hidden. Do those models on its own, do they induce these latent embedding spaces that capture some form of geometry or structural similarities between the samples, such as proteins that we are embedding, and whether those geometries are relate to the geometries of the latent space that graph neural networks generate based on protein interaction networks rather than primary sequences. And the interesting answer to that, by analyzing over 75 existing NLP-derived language models, is that there are indeed gaps between thinking of large language models that are developed for NLP and using them directly on more general biological sequences. And we can mitigate those gaps by introducing and developing new kinds of objectives. And here in specific, an objective that we introduce is that of structure-inducing objective that combines the language modeling objectives with objectives that are modeling the relational structure of samples, in this case being proteins based on protein interaction networks. And that kind of multimodal design then leads to improved performance on predicting various tasks related to therapeutic science. And here I'm just highlighting one of them on the right slide, where we were interested in asking the question of whether one can predict protein therapeutic targets based on structure and sequence more accurately than based on that primary sequence NLP language model alone or structural protein interaction network alone. Which creates an interesting opportunity of, well, can we first, let me just go back. Can we first enhance and augment prime KG knowledge graph and make it multimodal in the sense that it will combine both the relational structure of knowledge graph with the biological sequences? And second, can we actually also consider NLP-based language models in order to not make inference on the prime KG itself, but allow users to describe phenotypes and diseases more broadly, so that we are not limited only to reasoning about diseases that are explicit nodes in the knowledge graph. So one of the ongoing work that we are excited about here in my group is that of leveraging large language models to be able to predict disease states and phenotypes in a more universal manner, where domain experts now not only can go and interact with an AI model through explanations and predictions, but they actually can actively go and describe a phenotype and disease of interest in the form of plain text English and use it as a paragraph that then is processed by a text-based LLM. And that model, that text-based LLM, is then matched with an AI predictor, such as TXGNN, in order to produce priority lists of proteins, produce priority lists of drug candidates. The exciting consequence and implication of this work is that it does mean that we are not necessarily limited anymore to be making predictions around diseases that exist as explicit nodes in the knowledge graph, which are always diseases that exist in medical ontologies, the disease in medical vocabularies, such as ICD or Mondo disease ontology and so on. And so that means that it's possible to start thinking about out-of-vocabulary description of diseases and phenotypes instead of being a priori limited to only those phenotypic and disease concepts that exist in vocabularies. So all that work and this specific set of models that I've been describing so far are part of a broader initiative that my group is leading called Therapeutics Data Commons. And within Therapeutic Data Commons, we are releasing these various data sets, including PrimeKG and various models, including TXGNN, as part of the broader ecosystem that serves as a meeting point between, on one end, biomedical scientists who really know what are the critical challenges and barriers and bottlenecks across stages of drug discovery and development and across therapeutic modalities. And on the other end, AI scientists who can define and design strong machine learning models once those problems and drug discovery problems and tasks are defined meaningfully. And the overarching goal of Therapeutics Commons is to facilitate algorithmic innovation and advances in therapeutic science with a major focus on understanding what algorithms work best for a specific type of molecules and for a specific therapeutic use cases. And as I said, Therapeutic Data Commons is an ecosystem that contains pre-specified AI tasks and curated benchmarks. It provides leaderboards with best-in-class models for certain therapeutic tasks, and these pre-trained language models for key tasks that allow users to actually interact with the model and describe their specific phenotypes in diseases in plain language. And then models such as TXGNN and other interactive models that one can probe through web interfaces or API and interact with predictions made by the models. There are numerous compelling uses that my group has developed, as well as others, on top of Therapeutic Commons. In this talk, TXGNN has focused mainly around prediction of therapeutic use and zero-shot drug repurposing, but other models and support in Therapeutics Commons exist for tasks related to predicting properties of molecules and optimizing molecular structures, or modeling binding between therapeutic candidates and also candidate therapeutic targets from the structural or sequence-based view. And all is publicly available at tdcommons.ai, so those who are interested, we would love to have you join our community. So this is the first part of my talk. The second part is much, much shorter. I'm not sure how I'm on the time. 4:52. Okay, that's great. I have, let's say, 5 more minutes for the second part, which is shorter. But if I run out of time, at least I want to go to the motivation for this work, and then I'll point you to a preprint with the model. So far we talked about knowledge graphs with the AI that was and multimodal models that can process therapeutic tasks across a broad range of diseases. In the next part, we are interested in models that are concerned with and tackling a different challenge, which is that of contextualization. And by contextualization, I really mean models that can dynamically adjust their outputs based on biological context in which they operate. So why would we even want to do that? And what does that contextualization mean? Let me illustrate that with an example. Let's for a moment look at the word apple. So apple is a polysemic word. If you just see the word apple being written down, its meaning really depends on the context within which that word appears. You can grow an apple, you can buy an apple, and that meaning is resolved via sentence context. So we can look at now a gene, say H2AF hex gene, and this is a pleiotropic gene. So what this means is that the function that this gene performs on a cell, again, also depends on a context. But now this context is given by the context of, by the cellular context within which the gene acts. And the function of the gene would change with the context. So this is not only a single example, it's really common and widely understood from the biological standpoint that the roles or functions that the genes and gene products can have in different biological contexts can vary. And can vary significantly across the biological context in a cell, and even more so across health and disease states. The gap, however, between biology and existing machine learning approaches, specifically deep learning models, is that existing methods mainly produce context-free representations. In the very simple way, the way you can think of existing models is that existing models typically provide one embedding per gene or one embedding per protein. Whereas in fact, we might be interested in having a number of embeddings or representations per protein, where each of those embeddings would be specialized to a specific cell type or other biological context depending on their definition. And so we have recently developed a model that's called Pinnacle, which is a contextual graph neural network. It's a graph neural network that is trained on a data set of 156 protein networks from 156 biological contexts, which are cell types that we extracted from the Tabula sapiens human single cell atlas. And then with these contextualized protein networks, the model called Pinnacle is trained on those networks to provide contextualized representations of proteins. So the way how you can think of it is that those protein networks across 156 cell type contexts that span a broad range of tissues are fed into a graph neural network model that's optimized through a self-supervised link prediction objective. And the end result of that is that we have a suite or a set of embeddings for every protein, and those embeddings are specialized to biological contexts in which the underlying genes encoding for those proteins act. That can be used for numerous downstream applications, and I'm very happy to point you to the paper where we discuss and consider all those. And I will jump to the final slide here and wrap up my talk for today. So today we talked about two key capabilities that I think create new opportunities for the use of foundation and general purpose models in biomedical research. The first being that of learning on knowledge graphs and multimodal data that can process a vast array of tasks in a unified manner. And the second big opportunity is that of developing models that can be easily adapted and contextualized to a broad suite of biological contexts through the model Pinnacle and models of that kind that we are increasingly seeing being developed in the field that can produce contextualized predictions. All that would not be possible without a group of wonderful and fantastic students from undergraduates all the way to postdocs and research fellows that I'm working with and that are leading this research. And our AI models, data sets, papers are publicly available. You can go and see them through our lab website as well as through Therapeutic Data Commons or a separate AI for Science initiative that's a broader community that we are engaging in. Thank you for listening. I'm happy to take any questions in the time we have.
Thank you very much, Marinka, for a wonderful talk. So, questions. I can run around, and also you could please just come up here as well. That would be actually even easier. Oops. I'll give you one up, and then maybe you can also help run around as well, if this one works. Actually, it doesn't.
Hi, thanks for the talk. I wanted to ask about, for the TXGNN, you showed the interface and you have these drug embeddings. And I was wondering if you'd looked at how relationships between those, or distances between the drug embeddings, if they correlate at all with actual chemical properties of the drugs and their structures.
Yeah, so that's a great question. We have indeed done those tests where we have correlated drug embeddings with information on their structural properties, including SMILES fingerprints, selfies fingerprints, some of the other structurally derived embeddings of molecules that are generated based on 2D or 3D structure. And we have seen a strong correlation between similarity of embeddings derived from the knowledge graph and those from the structural models. Another comment that I can make is that interestingly, for the Pinnacle model, which is that contextualized models, we took contextualized protein embeddings, and we combined them with state-of-the-art structural embeddings of proteins generated by models such as Massif or models such as AlphaFold, GearNet, and other geometric deep learning models that are trained on protein structures. And what we are seeing that by doing a simple aggregation through concatenating protein embeddings based on structure with embeddings that capture this cell type specific information. When we select the cell type context for, in particular, PDL1, that is important target for immuno-oncology, and concatenate the structural embedding of PDL1 with that of contextual embedding of PDL1 from immune-related cell types, we can much more better accuracy identify binding partners of PDL1 than what would be possible if that contextual information would not be available. So what this shows is that in many ways, these different data modalities, specifically around molecules, is complementary. And I would not be arguing here that it's always good to consider either all the sequence or just the structural information, but considering all of them in a productive manner is probably what we ultimately want to do.
Yeah, I guess it would even be interesting to see where they differ, right? If they have the same contextual things and they're working on the same diseases but are very different structurally, that'd be super interesting to see too.
Yeah, so that's a great question. I didn't have time to go and show results specifically for that question because we can then ask what are the cell types and cell type contexts that are most predictive or least predictive of a specific question. So here we worked with our collaborators from Mass General on protein targets that are being examined for IBD diseases, and we were interested in knowing if there are certain cell types that are most predictive of targets that are currently being investigated in clinical trials that are being run by them. And we are indeed seeing that most predictive cell types are those that they would also expect to see at the very top of being involved in the mechanism of action for the drug that's being considered here.
Yeah, thank you.
Great question. So first, these knowledge graphs are highly incomplete and sparse. And that's that sparseness and incompleteness, the sparsity or the missing data is very challenging to handle because the data is not missing at random. In fact, what I mean by that is that the incompleteness in the knowledge graph is sometimes due to limitations of experimental platforms in being able to reliably probe interactions involving a specific protein. And even more so, in a separate study, we were interested in knowing whether the completeness of knowledge graphs can correlate correlate with kind of non-biological information, and in fact, it correlates with NIH funding cycles, which is in some way it's not surprising, because simply the volume of the data, which in the simplest case would be the density of the edges in the knowledge graph, can indicate not necessarily biological importance of a specific molecule in terms of just how many interactions does it have, but simply how easy it is, or how strongly community is interested in that specific molecule, and then profiling it experimentally. So there are numerous analysis that we have done to mitigate those effects that relate to processing and normalizing graph neural network with respect to the density of edges within the neighborhood of specific nodes, so that we can address for these challenges of data incompleteness in the knowledge graph. We're seeing the robustness of the model to be much better than that of more classic network analysis techniques that are based on shortest path calculation and characterization, where those mechanistic rules are incredibly sensitive to incomplete data, because simply the length of the shortest path between two nodes in the knowledge graph can change considerably by a single edge missing. And so here is actually where graph neural network models perform more robustly than some of graph theoretic techniques. Then the other question was, which of the data modalities would we like to have the most well represented in the knowledge graph? I would say that large-scale single central transcriptomic data sets on matched healthy and disease states across a large number of diseases would be just an incredible resource to have. And so one of our kind of ongoing efforts really is on that of matching TXGNN with Pinnacle in order to be able to consider this cellular context when making predictions that are made by the model.
Thanks, Marinka. I wanted to ask, when you build these these knowledge graphs, are you using curated data sets? Mm-hmm. And have you thought about using LLMs or other NLP techniques to mine these relationships directly from literature?
Yes, absolutely. That's a great question, and it's concerned with the problem of inferring or constructing the knowledge graphs. So the prime KG that is described here comes from curated repositories of data that is being directly experimentally generated. It is possible, and the research community is actually quite working across a broad range of applications that are concerned with completing knowledge graphs by predicting, leveraging the structure of incomplete knowledge graphs to predict new links and new edges in the knowledge graph. And you can think of those new edges and relations as being the hypothesis, but you can also think of them as actually part of the data pre-processing that can help you potentially complete the knowledge graph in a way that can help downstream machine learning models make better prediction. Our reasoning for this project was that we were specifically interested in using experimentally derived data, or data where we can go back to a primary experiment or primary research publication behind each and every edge in the knowledge graph that the model relied on for making prediction. And we kind of mitigate this problem of incompleteness and data sparsity to zero-shot learning, instead of a priori trying to fill in the knowledge graph and then introducing additional biases that would be very hard to account for in downstream models.
Thank you.
Okay, thank you very much. I think since it's time, let's maybe take more questions directly. Is that okay? You could just find Marinka. Okay, so thank you very much, Marinka.
Yeah, thank you for having me.